Now that we've covered the concepts regarding respiration, we're going to move into a lab. And in this lab, we're going to be calculating reaction rate, or the rate of respiration. Remember that respiration is just a series of reactions, and these reactions can happen at a faster rate or at a slower rate. In the biochemistry unit, we discussed that there's two ways that we can measure reaction rate. We can either measure the disappearance of some of the reactants, or we're measuring how much these molecules are being used up, or we can also measure the formation of products. Or we're looking at the change in products, and then we're going to take that and divide it by time. So this shows you the two different ways that we can calculate rate. And I want to make sure that you understand that we're going to use the disappearance of reactants over time to help us gauge respiration rate. Specifically, we're going to look at the disappearance or the use of oxygen over time. So we're going to see the change in oxygen levels, or the change in that reactant, over a period of time. Now, to measure oxygen consumption is a little tricky. So we're going to use a device that's called a respirometer. I've included some pictures of some respirometers, and you need to become familiar with these pictures and this setup because you're going to be creating a similar setup in this lab. So these are the respirometers, and one thing that we're going to do with respirometers after we've set them up is that we're going to submerge them in water. So let's show this respirometer is underwater. Whenever we do that, water is going to move into this pipette, but eventually it's going to stop. So let's show that water has moved in and it has stopped about right there. And it stops whenever the pressure outside equals the pressure of the respirometer. And where this water level stops is going to be our initial measurement that we're going to take. So we're going to indicate where this water has stopped, and these pipettes we're using are graduated. So we might see that it has stopped at 0.9 milliliters. Now let's take a look at the contents of this first respirometer, and we have some germinating seeds in there. And one thing that we're going to learn from this experiment is that germinating seeds require a lot of ATP because tissues are starting to develop in this plant embryo, which is a seed. And anytime we have development or growth, it takes a considerable amount of ATP. So we should see a high rate of respiration in these germinating seeds. And remember, in respiration, oxygen is being utilized. So we've trapped some gas in the respirometer whenever we've sealed it up. So there's oxygen in the respirometers, and the cells are going to be utilizing that oxygen as they're doing respiration, and they're also going to be producing CO2. If you think about it, if we're using molecules of gas and we're producing molecules of gas, then there would be no pressure change in this respirometer. And if there's no pressure change, then we're not going to see any movement of the water into the pipette. And that is why we have to use a substance called potassium hydroxide in the respirometers. And you can see here at the bottom of your respirometers, you're going to have a cotton ball and you're going to have it soaked with potassium hydroxide. Now, what potassium hydroxide is going to do is it reacts with the CO2 gas, forming a solid. And because you're changing the volume of the gas in this closed system, so if volume is going to go down, then what that does is it actually decreases the pressure in the respirometer. And if pressure is dropping and the outside pressure is staying constant, then the water is going to eventually move down that pipette towards the respirometer as these cells are utilizing oxygen. So let's indicate that removing the CO2 that's produced in respiration is going to cause that pressure to drop in the respirometer. So let's see how we're going to calculate rate from this information. Remember, rate is the change in something over the change in time. And if our initial water position in the pipette, like I indicated right here, is 0.9 milliliters, and after we let the respirometer sit for 30 minutes, we can see that the final water position is 0.5. Then our change is 0.4 milliliters. And that means that these cells have consumed 0.4 milliliters of oxygen. So that's our change, is 0.4 milliliters of oxygen, and that happened in 30 minutes. So that gives us about 0.013 milliliters of oxygen is being consumed per minute by those cells. And I have another respirometer here, and I have some data from it. So take a second to pause and see if you can calculate the respiration rate for the cells that are in the second respirometer. So again, we're getting a change of 0.15 milliliters of oxygen over time. The time was 30 minutes, and that's going to give us a respiration rate of 0.005 milliliters of oxygen per minute. Remember, if you are working a problem and it's part of an essay or a short answer, you have to indicate the units, because even if you get the correct answer, if you put the wrong units down, then you're not going to get a point for that. Whenever you're working a problem and it's a grid-in, you don't indicate units. So that's a situation where you don't have to show the units, but it is important that you show units on a short answer question. Now the idea in this lab is to measure this change, the movement of water, over a certain period of time. And so we're going to set up respirometers. We'll submerge them in water, and then every 5 minutes you're going to take measurements of this water level. So initially the water is going to move in, and then as respiration is occurring, this water level should continue to move in. And we have numbers on the pipette, and we're going to record those measurements. Now that we know that we can measure oxygen consumption of organisms, then we can set up these respirometers and test different independent variables. In our first experiment, we're going to see how the respiration rate differs in germinating versus non-germinating tissues. We can also look to see how respiration rate differs in endotherms and ectotherms. We can see how temperature affects respiration rate, and we could even compare the respiration rate of plants versus animals. So keep in mind, if you have a pet or you have a very small critter that you think can fit into a respirometer, we can actually measure the oxygen consumption of that organism over time. And when you design your independent lab, then you maybe can utilize that organism in your test. No one likes to hear that when we do a lab, we're going to have some issues with it, and that's going to be the focus on this page. But I'm going to address how we're going to fix these issues that will come up in the lab. So first of all, whenever we're loading up our respirometers here, we're going to put in some cotton and soak it with the KOH. We're going to put some non-absorbent cotton on top of that to kind of insulate the living tissue from that KOH, because that KOH is toxic to cells. And then we're going to put 10 germinating bean seeds in there. First, we're going to figure out the volume of those bean seeds. Now, as you can imagine, if we put 10 bean seeds in this one, then we also need to put 10 bean seeds in the second one. They're just going to be non-germinating bean seeds. So we have to keep the number of beans the same. The problem is when we germinate our bean seeds, we're going to wrap them in wet paper towels, and those bean seeds are going to absorb some of the water and start to germinate. So if you notice, these germinating bean seeds are much larger in volume compared to the smaller non-germinating bean seeds. So we also have to keep the volume of the contents the same in the respirometers. However, we just indicated that our 10 germinating beans are going to be larger than our 10 non-germinating beans. So in respirometer 2, we're going to make up the volume difference by adding some beads. These are glass beads that we're going to put in the respirometer. You can see them right here. So if we have a volume of 30 mils in that first respirometer, but we only have a volume of 20 mils when we measure out the non-germinating seeds, then we're going to have to add 10 milliliters worth of beads to that respirometer to make the difference up to 30. The second issue has to do with a change in temperature. Remember I said we were going to submerge our respirometers in a water bath. Well, the fact that they're in a water bath is going to try to help us maintain a constant temperature. But if the temperature of the respirometers or that water bath changes, then we're going to have movement of the water levels. Remember, water is going to move into the pipettes here to a certain position. This water level will move towards the respirometer or outwards depending on a change in temperature. But we do not want the water level to move due to a temperature change. We just want the water level to move due to oxygen consumption due to respiration by these seeds. So we don't want temperature to be a factor affecting our respiration data. So to help figure out if there is any change in the respirometers due to temperature, then we're going to set up a control group. And our control group is going to look like this. It's going to be a respirometer that just contains beads at the same volume as the other contents of the other respirometers. Beads do not perform respiration, so we should not see any change in the water level in the pipette. So water is going to move into the pipette to a certain position, and then it should stop there once we've allowed time for the respirometer to equilibrate with the bath water. And if there's any change in temperature, then we will see that water level move. If we see a drop in temperature of the bath water, then what's going to happen, we're going to have that initial water level there. If the gas that's in the respirometer cools off, then those gas molecules get closer together. So if there's a drop in temperature, that is actually going to drop the pressure of the gas in the respirometer. And if pressure drops in the respirometer compared to the outside pressure, then water is going to move in towards the respirometers. So if temperature drops, pressure will drop, and water is going to move inward. If the bath water increases in temperature, if someone's hands are in the bath water, then it can lead to an increase in temperature of that bath water. So if temperature goes up, then these gas molecules have more molecular motion. And with more motion, there's going to be a higher pressure in that respirometer. So pressure is going to increase in the respirometer. And if this was the initial position of the water, then a higher pressure in the respirometer would push that water level out. So water would be forced out or away from the respirometer. So it's important that whenever you start your lab, and you're going to have two different water baths, one that's at room temperature, which we will set up 2 days before class to make sure that that water is at room temperature, and then you'll have another one that is at 10 degrees Celsius. It's really important for that person whose job it is to maintain that temperature of 10 degrees Celsius is doing a good job. Otherwise, you're going to get lots of fluctuations in the respirometer with the beads, and you're going to have to do some math to correct your data. We want our data to show just the change due to oxygen consumption and not the change due to temperature. Here I have a table, and this is going to be similar to the table that you have on your Respiration Lab. And I already have some hypothetical data that could be collected from this Respiration Lab. Here we have germinating beans that we put in a respirometer, and the water is going to move into the respirometer, and it's going to eventually stop at a measurement. And so it stopped at 0.9. And then over time, the water level is going to move into the pipette towards the respirometer, and we're going to then write down these new values. Ultimately, this is what we're trying to figure out in this lab. We're trying to calculate the respiration rate for these germinating bean seeds. And to calculate rate, it's the change in something. In this case, it's the change in O2, or how much oxygen they consume in a period of time. Well, that's what these values are telling you, is how much oxygen that got consumed in 20 minutes. We had a change of 0.20 milliliters of oxygen. That's how much they took in 20 minutes. Now, I wish we could just plug those in and calculate rate, but we have a problem. If you take a look at the beads started out at 0.85, but the water level changed. The beads ended up changing or dropping negative 0.02. That negative means that they dropped. They went down from 0.85 to 0.83. Now, it's important that if they drop, you put a negative there. Because when you see a negative in math class, this means to subtract, or this means to take away. Now, the reason why the beads could have changed is because there was a pressure change in the room, or maybe there was a temperature change in the room. So what that means is our bean seeds really did not take in 0.20 milliliters of oxygen. Part of that change was due to a pressure change or maybe a temperature change in the room. So we have to correct that change in oxygen. So we're going to take away 0.02 from this value. So to correct this, it's 0.20 milliliters. I'm going to take away 0.02 milliliters. So really, they just took in 0.18 milliliters of oxygen. Now we can calculate rate. So our rate is 0.18 milliliters of oxygen, and divide it by the time, and that's going to be 20 minutes. And then we're going to get a value there, whatever that number is going to be. Now, we're having a decrease in oxygen. Our water level is decreasing. Ultimately, if you graph data points and did your change in something or change in time, you're going to see that this value is going to end up becoming, it's going to end up being a negative number because we're having a decrease in oxygen or a decrease in the water level. Now, let's take a look at the next page and work through some problems where you're going to have to correct the change in volume. Here we have non-germinating seeds, and we can see that there's been a change in the water position of the respirometers. And so our change in volume is 0.16 milliliters. So it dropped 0.16 milliliters. And it says that our beads also change as well. So we're going to have to correct this value. It said the bead water level moves inward toward the respirometer. So again, we're seeing it drop, negative 0.03 milliliters. This tells us, this negative means that we're going to have to take away from our initial volume. So the corrected volume is actually 0.16 milliliters, take away 0.03 milliliters. So the real change in volume is 0.13 milliliters of oxygen was really what was consumed. So to calculate rate, 0.13 milliliters of oxygen. Let's assume this took place over 20 minutes, and then we're going to have a rate. And again, this would end up being a negative value. Question 2 says, the non-germinating seed water level moves, so it drops 0.95 to 0.85. So my change in volume is 0.05 milliliters. Now, it says that the bead water level, it moved out 0.02. So let's explain what that would look like in your respirometer. Let's draw a respirometer. Let's put beans inside of it. Not beans, but beans. And let's put our pipette here. And remember, when we submerge these, water is going to move in until the pressure outside equals the pressure inside. So let's say our water level starts right here, and it stops right there. So at time 0, we are at, let's say it moved into like 0.9. I'm just going to use something easy, 0.9 milliliters, and that's where it stopped. But after 20 minutes, this water level has moved this way out, away from the respirometer. So now it's here. So let's call that, put that one there. At time 20, the respirometer is at 0.92. So you can see that we've had a change. Let's put a triangle there. Our change was plus 0.02 milliliters. If we have a plus there, that's going to mean that we need to add. So we're going to correct the change, 0.05 milliliters. We're going to add 0.02 milliliters. And that gives us the corrected change in oxygen is 0.07 milliliters. So we had really a drop, a negative, a drop of 0.07 milliliters in 20 minutes, and that's going to give us our rate. Again, this is going to be a negative number simply because oxygen levels are decreasing. So what you just did is you looked at data from the table and you calculated respiration rate. That's one thing you're going to have to be able to do after finishing this lab. Another thing that you're going to have to be able to do is calculate respiration rate if they give you the data from the lab, but they do it in graph form. Now, this is a graph that I took off the internet. So this is the data from AP students somewhere else that were doing this lab with the beams and in the respirometers. But this is an incredibly poorly labeled graph. Let's fix it. This 0.95 doesn't really mean oxygen consumption. It's actually more the water level. Okay, or we could even say the volume of the respirometer. It started at 0.95 milliliters. And then we would miss points here because they didn't put the units. In this case, it's minutes. So let's calculate the rate of respiration of the germinating peas. So they're just graphing the water level change over time. So let's just pay attention to the green line for the germinating peas and ignore all the rest. So again, rate is the change in Y, or my change in the water level, or change in oxygen, over my change in time. So let's start out with my change in Y. It starts at 0.95 milliliters, and it goes down to all the way here, 0.61 milliliters. And the time at 0.95 was 0 minutes, and at 0.61 we were at 30 minutes. This is also going to show you that this is where this negative value is going to come from. So let's do the math here. So 0.95 minus 0.61 gives us 0.34 milliliters divided by negative 30 minutes. And that's going to give our respiration rate. Again, that number is going to be a negative. Now, in the past, I had students graph the water level or the reading of the pipette over time and then calculate rate from the slope of the line, which is exactly just what you did here. So you're probably thinking, oh, okay, so I would plot 0.9 and 0.85 and 0.83, and then I'd calculate the slope of the line. But there's a problem here because these beads are showing us that there was a change in temperature or pressure. And so our water levels actually shouldn't really be at 0.9 and 0.85. We would have to correct those. I'm just going to write in some values here. These are the corrected water positions when you take in consideration that some of the change wasn't due to oxygen consumption, it was due to maybe a temperature change. So this would be 0.82 milliliters, this is 0.77 milliliters. These are the numbers that you would put on the graph, like I just showed you on the previous page. Now, to show you how to correct and figure out these values, it's incredibly complicated. And students usually just kind of stop focusing on the big picture. I just need you to calculate respiration rate. What is my change in oxygen over my change in time? So on your lab, I'm not going to have you fill in these values and then graph these data points over time. But we still can calculate respiration rate doing what we just did at the very beginning when we started talking about the data that was in that table. To summarize, ultimately, what you have to be able to do is look at a graph of the data from this type of lab and calculate rate from a graph, or look at the data in a table and calculate respiration rate from the data in the table. Those are the two things that I want you to focus on for this respiration lab. Now, I like this lab because it shows you that we can set up an experiment and we can test two different independent variables at one time. Lots of times in your lower grades, you're taught that you can only do one independent variable at a time, but you can set up many different IVs at one time that you want to test, and then you just have to pick which treatments to compare to help you draw a conclusion. So I've got these two squares here, and they're indicating these water baths that we're going to set up. Remember, one is going to be set up at room temperature the day before the lab, and then we're going to establish one at 10 degrees Celsius. And so someone is going to be stirring ice into that water bath as soon as we get to class to try to get that water bath down to 10 degrees Celsius and trying to maintain it. And if you're not maintaining it, we're going to be able to see that when we look at the water position here in the respirometer that contains those beads, because it shouldn't change, but if there's a temperature change, then we're going to see a change in that water position. So if we take a look at the setup of our experiment here with these 6 respirometers, you can see one thing that we're going to change is the temperature. So how does temperature affect respiration rate? And then the second thing you see that we're changing is we're putting germinating seeds in one and non-germinating seeds in the other. So we're going to see how does germination affect respiration rate. So let's do some questions related to this experiment and the fact that we have 2 independent variables being tested. It says, what respirometers would you compare to determine if germination affects respiration rate in cells? So if we want to see if germination increases or decreases respiration, then we could either compare 1 versus 2, and I have them numbered for you for reference here. So we could either compare the results of those two respirometers, or we could compare 4 and 5, because we're just changing between 4 and 5 that we have germinating seeds in one and we have non-germinating seeds in the other, and we're keeping temperature constant because both of them are at 10 degrees Celsius. Question 2 says, what respirometers would you compare to determine if temperature affects respiration rate of germinating cells? So in this situation, our IV is temperature. So we have germinating cells, germinating seeds in respirator 1, and we also have germinating seeds at a different temperature in 4. So we would compare the results of those two respirometers. And then finally, question 3 says, what respirometers would you consult to determine if a change in temperature is affecting the results? And that is the purpose of our control group, the beads. So we would look at respirometer 3 to make sure that there is no temperature change in the bath that's at room temperature, and we also need to check respirometer 6. Now we're going to discuss different factors that are affecting respiration rate. One concept that you'll see in the respiration lab when we're comparing the respiration rates of the germinating versus the non-germinating beans is that with the germinating beans, they're utilizing more oxygen because they need more ATP because they're trying to grow. Growth and development of tissues takes a considerable amount of ATP. That is why as a teenager, you have a higher metabolic rate, you have a higher respiration rate than adults because you're trying to grow and it just requires more ATP for that process. Also, you are more active, so the higher the activity level, then the higher the rate of respiration. You have to make more ATP so your muscles can function, so therefore you're going to require more oxygen and glucose. Anytime you change the concentration of the reactants in a reaction, then that can alter rate. Referencing the equation for respiration, we know that oxygen and glucose are substrates, so let's indicate that. And we learned in the biochemistry unit that if we increase the concentration of the substrates, then reaction rate is going to go up, but eventually it's going to hit a maximum. And this is a maximum rate. This does not mean that the reaction is over because we have rate on the y-axis, not change in product over change in time. Even if we add more glucose or increase the concentration of glucose, those enzymes will become saturated, and adding more glucose is not going to allow reaction rate to increase. With oxygen, I want to indicate that we're not going to start back at 0, 0, because remember when we're out of oxygen, so 0 oxygen, we still have a low rate of respiration because anaerobic respiration is occurring. But again, eventually we've hit our maximum rate for respiration, and adding more oxygen is not going to cause the reaction to take place faster. Let's indicate the equation of respiration. Because I want to emphasize that increasing the concentration of the products will have no effect on rate. So let's put an X through there and indicate you have no effect on rate. Increasing temperature increases reaction rate. At a low temperature, we'll have a very low rate of reaction because there is less molecular motion and the enzymes are not colliding with the substrates as often. But as we increase temperature, reaction rate goes up, but eventually we've hit an optimal temperature, and past that point, so past this temperature, we know that denaturation is occurring. So let's write denatured enzymes because every reaction in respiration is facilitated by an enzyme. And we also talked about changing pH can alter reaction rate, and every enzyme has its optimal pH. So let's indicate for this one, let's say that it operates more in an acidic environment. So again, every reaction in these series of reactions of respiration requires an enzyme. And whenever the pH is too off from the optimal pH, then the enzyme can have a shape change. Here's a topic I've seen several times on released items. Endotherms and ectotherms are going to have different changes in respiration rates due to temperature. So take a second to read the example problem in this example question 1 and cover up these answers here because I want to see if you can come up with the correct answer. The big idea to understand here is that whenever there is a drop in temperature for the mouse, it looks like we have an increase in respiration rate. And you can see that because it consumed a lower amount of oxygen at a warmer temperature, but it consumed more oxygen at a colder temperature. The cricket data is showing something else. If there's a drop in temperature for the cricket, it looks like its respiration rate is dropping as well. This relationship happens in all endotherms, and this relationship happens in all ectotherms, and you have to be able to explain why. I decided to write out the explanations here so that you can see an example of a good answer to this question. So explain why the mouse at 10 degrees Celsius had a higher respiration rate than the mouse at 25 degrees Celsius. To answer this question, you have to remember that mammals, like mice, are endotherms. Can you think of another type of animal that is considered an endotherm besides a mammal? I'll give you a second to think about it. Birds are considered endotherms as well. They can regulate their body temperature. Now let's try to explain why whenever the temperature of the environment dropped, then the mouse had an increase in its respiration rate. So if there's a drop in temperature, so if the mouse is in a cold temperature like 10 degrees, then its body temperature is going to start to drop. But remember, endotherms are going to maintain homeostasis, a constant body temperature. So in response, you're going to see the mouse is going to shiver, and that requires ATP to contract those muscles. As ATP is used, more ATP has to be produced. And the fact that we are breaking down ATP, and also that we're using all these reactions in respiration to make more ATP, will also release heat, according to that second law of thermodynamics. The second law of thermodynamics states that when there's a transfer of energy, that it's never 100% efficient. Some of that energy is going to be lost as heat. So all these reactions that are taking place to contract muscles and to produce ATP through respiration, those reactions are going to transfer energy from one molecule to the next, but some of that energy is going to be lost as heat. And the fact that heat is being generated is going to help this mouse increase its body temperature to maintain it at a constant level. So we can see that respiration rate is going to increase due to the fact that more oxygen is needed to produce that ATP so that mouse can maintain homeostasis. To help us remember, let's indicate that the mouse is shivering, whereas this other mouse at room temperature is not. So it's not going to require as much ATP, thus as much oxygen, and it'll have a lower respiration rate. Let's take a look at the explanation for example 2. Why did respiration rate of a cricket decrease with a temperature decrease? Crickets are ectotherms, which means that they cannot regulate their body temperature internally. So if the environmental temperature drops, then so is the body temperature of the cricket. So all these reactions that are taking place now in the cricket are now taking place in an environment that's cooler, and we know that molecular motion is going to decrease, and so our reaction rates are going to drop. So respiration, which is just a series of reactions, is going to drop in rate as well. So if respiration is occurring at a slower rate, then that means there's going to be less oxygen that is needed. So that is why you see the cricket at a lower temperature is taking in less oxygen compared to this cricket here. So this cricket is warmer, and in warmer environments, reaction rates like those involved in respiration are going to be increased, and this one is colder. So reactions are taking place at a slower rate. Take a second to read through questions 3 and 4 and see if you can come up with the correct answer. Hopefully you determined that A is the correct answer. Because that mouse was trying to maintain homeostasis, it had a higher rate of ATP production because it was utilizing a lot of ATP as it was shivering. B is incorrect. As a matter of fact, the mouse at 10 degrees Celsius had a higher metabolic rate, so it was incorrect. C, the mice at 25 degrees weighed less. This is an example of you don't have that information, so that was probably an easy answer to throw out. And then the mice at 25 degrees Celsius were more active. Again, you weren't given any of that information, so that really couldn't be a plausible answer. Looking at question 4, the correct answer was C on this one. A, you'll never hear me talk about the difference in tissues between cricket and mice, so you're going to know to throw that one out. This one says enzyme activity decreases in mice as the temperature increases, and we know that enzyme activity in general, it increases as temperature increases. And then I think D is just an obvious throw out answer. Now we're going to get into a statistic that relates to what we just talked about with those crickets and the fact that whenever they dropped in body temperature, the reaction rate fell as well. What this Q10 value is it allows us to determine how much reaction rate is going to be changed by a change in temperature of 10 degrees. So think of those crickets, and if their body temperature drops 10 degrees, then we have some kind of indication of how much reaction rate is going to drop. I have some hints about Q10 values if you're ever asked to do this on an AP exam. Q10 values are usually between 2 and 3. So if you calculate the Q10 and it's off from 2 to 3, then you probably have done something wrong and you might retry. And second is if you look at this equation, so here is the equation for Q10, and this is what it looks like on the formula sheet. You can see here that we're going to have an exponent. But remember, you have a 4-function calculator that you're allowed to use on the AP exam. So you won't have that exponent button. You're not going to have an exponent of like 1.43. Because you don't have that exponent button, then whenever you simplify on an AP exam, you're probably going to see that they have made this a whole number. So then it's easy to plug in a 4-function calculator. So you're just going to divide these two numbers, and then if it's a whole number or exponent of 2, then you just times that number by itself. Or it's a exponent of 3, then you take this number right here and you just times it by itself 3 times. So let's work one of these problems, and I'm going to take a second to erase some of this. Over here is your definition of what all of these symbols mean. Basically, you have to read the problem and figure out which number goes with K2, which number goes with T2, and so on. Plug it into the equation and solve. So we have a goldfish, and it's in water at 17 degrees Celsius, and this is its respiration rate. And I'm probably going to pronounce this wrong, but the operculum is that flap that you see right behind the eyes of the fish that open and close. And that basically opens and closes so that a fish can draw water across its gills, and it can get oxygen from the water. So it's related to breathing or acquiring oxygen, kind of like we use our diaphragm to pull oxygen into our lungs. So it says that its respiration rate is 110. So these two go together. This is going to be our temperature 2, and this is the rate, and K is rate. So this would be our K2. Sometimes I like to call it R2 for rate, but we'll just go with K2 so you're not confused when you look back at these notes later. So that is the rate that goes with that temperature. Well, here's another temperature right here. So here is our T1, and here is then our K1, or that first rate. So let's go ahead and plug these into our equation here. So Q10 equals, our K2 is 110, K1 is 62, and then it's 10 divided by T2 is 17, minus T1, and T1 is 10. So what we're going to do first is we're going to do this. So we would take 10 divided by 7, and that gives us 1.43. So that's the exponent. And obviously this wouldn't be an exact problem that you would work on an AP exam because, again, you can only use that 4-function calculator. But I kind of have rights to giving you exponents like that because I know you can use your other calculators in class. And let's go ahead and simplify 110 divided by 62. That gives us 1.7. So basically we have 1.7, and the exponent is 1.43. It is 2.27, and you can see that checks out between 2 and 3, so we probably did this calculation correctly. I also could see that if you're given an example like this and you had to calculate Q10, they could also go on to ask you to explain why respiration rate drops whenever that fish was changed from 17 degrees Celsius to 10 degrees Celsius. And again, it's because they are ectotherms. So as their body temperature drops, then the reaction rates in their bodies are going to drop as well. Fish cannot regulate their body temperatures internally like those endotherms can. You might take a second to look at the next example and see if you can work this example. I want to show you that sometimes you can be given multiple temperatures and multiple rates. Basically, you just need to pick two. And if you want to call this your temperature 2 and this one your temperature 1, then this will be your K1 and this will be your K2. And you can ignore the last data points given to you there. Just go ahead and see if you can calculate that Q10 value. So plugging in those values, we have Q10 equals 22 is that first rate, is K2, 16 is K1, and for the exponent we have 10 divided by 21 minus 17. And again, that is T2 minus T1. And if we simplify in parentheses, that's 1.38. And for the exponent, we would get 2.5 because 10 divided by 4 is 2.5. When you plug that into the calculator, then that gives you a 2.21. And again, it checks out between 2 to 3. So we're probably calculating it correctly.